Hello friends, in the previous lectures we were talking about the pathophysiology of hypertension, and today we will start another series of lecture explaining about the pathophysiology of ulcer. But before starting the pathophysiology, let's talk about physiology of GIT. So first of all, here you can see, as you know, that there are basically 5 anatomical regions of GIT. The major portion of GIT is basically consists of 3 anatomical regions, that is fundus, body and antrum. But overall, GIT is made up of 5 anatomical regions: cardiac, fundus, body, antrum and pylorus. Cardiac is near the esophagus, but pylorus region is near the duodenum. So overall there are 5 regions. And if we talk about the wall of stomach, we will get to know that the wall of stomach is basically made up of 4 layers. The innermost layer is called as mucosa. So when the GIT is, when the stomach is empty, this mucosal layer contract to form folds and a rough surface. These fold and rough surface is called as rugae. It's called as rugae, and rugae is basically made up of small gastric pits, and in the gastric pits different types of cells are present. The gastric pits that are present in the fundus and body will be having different types of cells as compared to the gastric pits that are present in the antrum and pylorus. Okay, so here you can see I have made one gastric pit in large, so that we can talk about different types of cells present in the gastric pits. At the top, here you can see the most important cell, mucous cells or favular cells are present. These cells are basically responsible to secrete mucus that will protect the epithelial cells of stomach from acid. It will also release, this favular cell, they will also release bicarbonate, and these bicarbonate basically will neutralize the acidic nature of HCl. Okay. Other than favular cell, you can see columnar epithelial cells. Columnar epithelial cells basically present throughout the GIT. Our GIT is made up of columnar epithelial cells, but other than columnar epithelial cells, these some other secretory cells which include favular cells, and the most important cell which include parietal cells are present. Parietal cells are basically responsible for secretion of HCl, and because of this HCl, pH of stomach is always HD, it's approximately 2 to 5. Other than parietal cells, here you can see green in color enterochromaffin cells, and these cells are basically responsible for secretion of histamine. Okay, and at the last, chief cells are present. Chief cells will release pepsinogen. Pepsinogen is an inactive hormone, inactive enzyme, but when it will come in contact with HCl, it will get activated and will be converted into pepsin. Pepsin is basically responsible for digestion of proteins. This pepsin will convert proteins present in the food to amino acids. Now, let's talk about detail about these cells. As I have explained that favular cell will release mucus and bicarbonate, here you can see prostaglandin receptors are present on the favular cell. Prostaglandin is basically a membrane-derived mediator. Arachidonic acid, when COX enzyme will act on the arachidonic acid, it will be converted into prostaglandin. The detailed pathway will be explained when I will talk about the pathophysiology of ulcer due to NSAIDs. Okay, then detailed pathway will be explained. But here you get to know that prostaglandin will be having its receptor, and when this prostaglandin will act on its receptors present on the favular cells, mucous cell, it will increase. Positive sign is indicating that it will increase the secretion of mucus and bicarbonate ion. Okay. Here you can see prostaglandin have also its receptors present on the parietal cell, and when prostaglandin will act on its receptor present on parietal cell. It will decrease the secretion of acid from the parietal cell. Clear? Now come toward the parietal cell. On the parietal cell, two most important receptors, M3 receptor, which is a muscarinic receptor, and H2 receptor for histamine are present. When M3 receptor will be stimulated with the help of acetylcholine. Acetylcholine is a neurotransmitter of the parasympathetic nervous system that acts during rest and digest. While we are taking a rest, it means our parasympathetic nervous system is activated. And when the parasympathetic nervous system is activated, it means acetylcholine will be released. And when this acetylcholine will act on its M3 receptor, it will increase the secretion of HCl from the parietal cells. Okay. At the same time, here you can see H2 receptor for histamine. We know that enterochromaffin cell will release histamine. Again, acetylcholine will stimulate enterochromaffin cell to release histamine, and this release histamine, when it will act on H2 receptors of parietal cell, it will increase acid secretion again. It will increase acid secretion from the parietal cells. Well, here you can see there are some other receptor for the gastrin, receptor for somatostatin. This enterochromaffin cell is also having receptor for gastrin. So now let's talk about what are gastrin and what are somatostatin. In the while at the start of the lecture, I have explained that the gastric pits present in the fundus and body will be having different types of cell as compared to the gastric pit that are present in the antrum. Now let's talk about the gastric pit that is present in the antrum. Here you can see two important cells, G cell and D cell, are present in the gastric pit of antrum. These G cells will release gastrin. It will release gastrin. So, and gastrin will go and it will increase acid secretion. Gastrin will increase acid secretion by direct method or by indirect. It will increase acid secretion by direct or indirect pathway. By direct method means that this gastrin has its receptor present on the parietal cell. Here you can see that this gastrin will act on its receptor and increase secretion of acid. So this is the direct pathway in which gastrin is increasing the acid secretion. There is an indirect pathway. Here you can see gastrin is also having its receptor on the enterochromaffin cell, and when this gastrin will go and it will act on it, it will increase the histamine. It is not increasing the acid this time, it is increasing the histamine. But we know that histamine will ultimately increase the acid secretion when histamine will act on the H2 receptor on the parietal cells. Okay, so gastrin is basically increasing the acid secretion. Now point is, who is stimulating G cell to secrete the gastrin? Now let's move toward the chief cell to discuss this point. We know that chief cell will release pepsinogen, and to activate this pepsinogen, to get pepsinogen converted into pepsin, we need HCl. In the presence of HCl, this pepsinogen will be converted into pepsin, that is active form of pepsinogen, and this pepsin will digest protein and convert them into amino acids. Now these amino acids will go and they will stimulate G cell to secrete more gastrin. Now this gastrin will go, it will bind on its receptor and it will increase acid, and this acid will further activate the will further convert the pepsinogen into pepsin, and in this way this cycle will keep on going. So it means basically food or proteins or amino acids present in the stomach, they basically stimulate G cells to secrete the gastrin. They are basically amino acids that are telling the G cell to secrete more gastrin because we need digestion, we need HCl. Okay. Now let's talk about the somatostatin. Somatostatin will be secreted from the D cells. Somatostatin will be secreted from the D cell and it will inhibit the acid. Somatostatin will inhibit the acid release. Again, somatostatin will act by direct pathway and by indirect pathway. Somatostatin will act by direct pathway or by indirect pathway. In direct pathway, here you can see somatostatin is having its receptor present on the parietal cells, and when somatostatin will go and bind to its receptor, it will decrease. Here you can see negative sign. It will decrease the acid secretion from the parietal cell. So this was the direct pathway. Somatostatin can also reduce acid secretion by indirect pathway. Here you can see somatostatin is also having its receptor present on the G cell, and when somatostatin will bind on the G cell, it will inhibit, negative sign, it will inhibit the release of gastrin, and obviously we know that when gastrin secretion will be inhibited from the G cell, ultimately it will decrease the acid secretion either by direct or indirect pathway. It will decrease the acid secretion from the parietal cells. Okay, so basically somatostatin is decreasing the acid secretion. Now point is, who is stimulating the D cells to secrete the somatostatin? To answer this question, let me tell you, we know that normal pH of stomach is 2 to 5. Okay, or in some literature it is written in 1.5 to 3.5. Point is, it is acidic in nature. Is because of this HCl, stomach pH is acidic. Lower pH is basically indicating the acidic nature. But if HCl is keep on releasing because of gastrin, because of histamine, because of acetylcholine, if HCl is keep on releasing and the pH gets too low, or below 1, now this is very dangerous because it can cause corrosion of the cells. It can cause ulcer, it can cause damage the apical side of the cell, epithelial cell. So now what will happen? This low pH will stimulate the D cells to secrete the somatostatin. This low pH will stimulate the D cell to release the somatostatin. Now this somatostatin will be released and it will inhibit the further secretion of HCl because pH is already very acidic. So now we need to inhibit the acid secretion, and this action will be performed by the somatostatin. Okay, so this was all about the role of different cells present in the gastric pits of fundus and body or in the antrum. Okay, so now let's talk about some protective factors and destructive factors that are present in the stomach. Okay, that are present in the stomach. The protective factor include mucus and bicarbonate. Mucus, obviously, for example, here you can see this green color is the mucus, and these are the cell line. Mucus is basically protecting the cells from the acidic effect, from this harmful acidic effect of HCl. Okay. Similarly, bicarbonate ion will have tendency to neutralize the acid. So obviously they are basically considered as protective factors, and we know that secretion of bicarbonate and secretion of mucus can be enhanced with the help of prostaglandin. Furthermore, somatostatin will also come into the category of protective factor because we know that when the pH of stomach is too much acidic, somatostatin will play its role and it will inhibit the further acid secretion. So obviously we will place somatostatin in the category of protective factors. The other protective factor is resistance of apical side of cells from the acid. Here you can see this is the lumen of the stomach. Okay, and the D cell, the side of cell that is facing the lumen of stomach, they are called as, this side of cell is called as apical side, and opposite side of cell is called as basolateral side. This apical side of columnar epithelial cell naturally have tendency to resist acid to some extent. This apical side. So because of resistance of this apical side of cell against the acid will also be considered the protective factors. Will also be considered the protective factor. And the last one is blood flow to the mucus. Obviously, prostaglandin again play a very important role in the vasodilation and increasing the blood flow toward the GIT, or increasing the blood flow toward the mucus. When there will be more blood flow, obviously tendency of regeneration of damaged cell will increase. So blood flow toward the mucus will also be placed in the category of protective factors. Okay. Now let's talk about the destructive factor H. pylori. Helicobacter pylori is a bacteria that can cause ulcer. Detailed pathophysiology of this bacteria will be discussed in the later videos. I will explain how this bacteria will adhere and how it will cause toxicity, how it will damage the cell. All these mechanisms will be explained. Next, gastric ulcer. Obviously, if the release of acid from the parietal cell increase, obviously it can damage these epithelial cells. It can damage these epithelial cells because of any carcinoma, gastrinoma. If acid secretion increases, so it can cause damage to the epithelial cells. So we will place gastric acid into the destructive category. Pepsin. If there is high pepsin, obviously more protein will be converted into amino acids, and these amino acids will stimulate G cell to secrete more acid. And when there will be more gastrin, and when there will be more gastrin, obviously there will be more acid. Okay, so pepsin will be also placed in the category of destructive factor, because pepsin will convert protein into amino acid. Amino acid will stimulate G cells to secrete gastrin, and gastrin will further stimulate parietal cell to secrete acid. So we will place pepsin into the destructive factor category. And the last, NSAID. NSAID is also come into the category of destructive factor, because NSAID basically block these prostaglandin. NSAID basically block this cyclooxygenase enzyme, and when this cyclooxygenase enzyme will be inhibited, arachidonic acid conversion into prostaglandin will be inhibited. When there is no prostaglandin, obviously the secretion of mucus and bicarbonate ion will be decreased. Similarly, secretion of acid will also be increased, and when the secretion of mucus decreases, for example, when there is no mucus, obviously now this cell will come into direct contact of HCl, and now these epithelial cells will start damaging. Okay, so these are the destructive factors. It was all about the physiology of GIT, physiology of stomach. I hope you must like the lecture. Please don't forget to like and share this video, and don't forget to subscribe my channel and press the bell icon so that you may get notification for the future video. In the later videos, in tomorrow videos, I will talk about the detailed mechanism of synthesis and release of acid from these parietal cell, and then in next videos I will talk about the pathophysiology of ulcer because of H. pylori, because of gastric acid oversecretion, and because of NSAIDs. Thank you so much.